Yeah, actually, please do remind me if you notice that. Um, Last Tuesday, I never recorded the lab meeting, so I had to post up an older lab meeting. I think everything is correct in the older video, except for the dates, but everything else was the same material. All right, I think we're done here. Let me just, I already mentioned about the lab. Remember that you can turn in an extra study group. Click on this link here and it'll tell you what to do. You need to get it submitted by 11:59 p.m. this Saturday for 1 extra credit point. And remember, everything that's an extra credit is going to be out of 0 points because I don't want the points to add to your total grade. So if you get anything out of this, it'll be extra credit. I think that's it. Checking to see if there's anything else I should talk about. No, okay. Oh, here's one other thing. Only a few students have turned in an infectious disease research project. Please pick a project and then clear it with me because I want each of the projects to be separate, meaning I don't want two people taking the same project. And there has been one request where I told the students you have to pick another topic because your request has already been selected. So please clear it with me. If you don't clear it with me, you will be penalized. I might not knock off 100 points, but you have to clear it with me before you can choose that infectious disease project. All right. I think we talked about this slide. I don't have it marked. Did we talk about this slide? Organic compounds can be made by putting two monomers together to make the polymer, or specifically the dimer, but we add on the monomers to make a polymer. I don't think we've talked about this slide. Is that correct?
I don't recall it.
Okay. All right, so you can add two monomers by pulling off OH group from one monomer and a hydrogen group from the other monomer, and then bringing those OH and H together to form water. And so water comes out of this reaction. And this is a condensation reaction or a dehydration synthesis reaction. Both of those mean the same, where the monomer, which is just R, stand for some organic chemical group, then will be bonded together where the R will bind to the R, making the polymer. And you can add water to a polymer, such as right here, and then go backwards. This reaction is called hydrolysis. And then the water splits apart with the hydrogen going to one R and the OH going to the other R. And then you can get back the monomers. Any question about any of that? We use dehydration synthesis all the time for making our macromolecules, like when you're building a protein or a carbohydrate. And of course, when you're eating those molecules and you're digesting them, we use hydrolysis all the time to break down the molecule. And looking at it from a little different perspective, dehydration synthesis, you add on a monomer, OH comes off of the monomer, hydrogen off of the polymer, and then the monomer is bounded to the rest of the polymer, and then water is synthesized. Hydrolysis is the reverse, where we add water, which breaks the bond between this part of the polymer and that part breaking off the monomer. And the water splits apart, the OH going to the monomer and the hydrogen to the polymer. So hydrolysis and condensation synthesis are the reverse reactions to each other. So let's talk about the 4 biological molecules. Carbohydrates is the first one we're going to talk about. That includes molecules such as simple sugars, starch, and other complex carbohydrates. They are important as energy sources for organisms, for cells, as well as for the structure, and they're important in nucleic acids. Carbohydrates consist of carbon, hydrogen, and oxygen, but just not in any formulation. They have the general formula that for each carbon, there's 2 hydrogens and 1 oxygen. And then you just repeat that for the carbohydrate. Like glucose, for example, would be CH2O repeated 6 times, or C6H12O6. Any questions about any of that? So carbohydrates have this specific formula. Carbohydrates include the simple sugars or strings of sugars put together to make the complex carbohydrate. You can classify carbohydrates into 3 major groups based on their size. The smallest are the monosaccharides. That would be something like glucose and fructose. And then 2 of the monosaccharides are disaccharides. That would be table sugar or sucrose. I almost lost it there. And then the many monosaccharides put together are the polysaccharides. And these are the complex carbohydrates, such as starch and glycogen and cellulose. So let's talk a little bit about the monosaccharides. These are simple sugars. All of them range in size from 3 to 7 carbon atoms, but most of the simple sugars, or the monosaccharides, are either 5 carbons or 6 carbons. They're all sweet molecules and water soluble, meaning they will dissolve in water. The disaccharides are just 2 of the monosaccharides joined together in dehydration synthesis. And for example, we have glucose, a monosaccharide here, and then fructose, a monosaccharide here. And in dehydration synthesis, you pull out the water, and then you link the 2 monosaccharides together right here. And hydrolysis is the reverse. You add water, which is not shown, water here, breaking that bond, and then making the monosaccharides. Please understand that when we're talking about the carbohydrates, they can be drawn in a linear fashion or in a ring fashion. Here we're saying the ring fashion, and when we draw it in a ring fashion, wherever there's a point, like right there and right there, that's understood to be a carbon. If it is not a carbon, they have to put in the abbreviation for the element. It's like right here, it's oxygen. Any question about any of that? The sugars can be in a linear form or in the circular form, depending on how the molecule is structured. Fructose, if it's in water, most of it is in the circular form. And then glucose, if it is dry, out of water obviously, most of it is in the linear form. The complex carbohydrates are called polysaccharides. They consist of many monosaccharides hooked together, one after the other. The polysaccharides can be straight chained, like starch, or can be branch chained. And I'll show you a picture of that. Glycogen is an example. Some examples of the polysaccharides: starch, glycogen, dextran, cellulose. All are polymers of glucose. They can be covalently bonded differently, like cellulose and starch have different bonds. And there's some starchy foods. You don't need to know this table. I'm just showing you the different polysaccharides. As I mentioned, starch is mostly a linear chain molecule having this chemical form, one glucose repeating after the other. Glycogen is a similar molecule, but it's in a branching chain. Cellulose is similar in the sense that it is the same glucose repeating one after the other, but it's a different chemical bond between the glucose. And the cellulose also has hydrogen bonding, which it's not shown very well here. Let's see if I can blow that up. See how this is a broken chain between the glucose there, and then the covalent bond is a blue line there. So the glucose is bonded together in cellulose by a covalent bond, but between those glucose, the next row of glucose is actually hydrogen bonded to the cellulose chain. Anyways, you don't need to know this. I just wanted to point out some of the different polysaccharides and how they look. All right, any question about the carbohydrates? The first of the 4 biological molecules. If not, let's work on the lipids, the second of the biological molecules. Lipids consist of carbon, hydrogen, and oxygen. And although all lipids do have some oxygen, most lipids, if not all lipids, have very little oxygen. So most lipids are mostly carbon and hydrogen. Most lipids are nonpolar and insoluble in water. We call them hydrophobic. There is an exception to that, and we'll talk about that later. Phospholipids are an exception, and they're an unusual lipid in that they are not hydrophobic. And part of the molecule is not nonpolar. We'll talk about that in a little bit. But all the other lipids are nonpolar and insoluble in water. Lipids are structural molecules in our cells and in organisms, and they're important in energy storage roles. Lipids are the primary components of the cell membranes, and when you're storing energy or molecules, like when you eat more than you can use or burn, you'll store it down as a lipid. Even if you eat lots of carbohydrates and lots of protein, your body won't store it after a while. I mean, it will store a little bit as carbohydrate, but if you eat more than you need, we'll convert it to a lipid and then store it as a lipid. There are 3 categories of lipids: simple lipids, complex lipids, and steroids. Let's talk about the simple lipids first. They're made up of fatty acids as one component. Fatty acids consist of a fairly long chain of carbons held together by the carbon bonding to the carbon. And there is one carboxyl group, the COOH group, at the end right here. Most other locations there are hydrogen bond to the carbon. For a saturated fatty acid, we have the maximum hydrogens possible on the carbon. And the carbons are bound together with a single carbon bond, a single covalent bond. That's a saturated fatty acid. For an unsaturated fatty acid, you have at least one double carbon bond in the carbon linkage. And that creates just one, the entire molecule, unsaturated fatty acid. You can find unsaturated fatty acids and fatty acids in general in oils such as corn oil. Wherever there's a unsaturated fatty acid linkage between the carbons, there will be a kink in the molecule. Let me see if I can blow this up. So right here we have the unsaturated link between the carbon, making this entire molecule unsaturated because we only need one to be unsaturated. Then the molecule is called an unsaturated molecule. But wherever there's the unsaturated link, there's a kink in this fatty acid. And so instead of being straight like this, it then bends right there. Any question about any of that? All right, for the simple lipids, they are a combination of fatty acids hooked onto a glycerol molecule. Here, let me show you the glycerol. It's a simple 3-carbon molecule, and the glycerol binds to the fatty acid in dehydration synthesis. The water comes off, the OH comes off of the carboxyl group of the fatty acid, hydrogen comes off the glycerol, and then you link the glycerol, that O, to the C of the carbon. So simple lipids are called fats or triglycerides, and they're called triglycerides because they have 3 fatty acids, 1, 2, 3, bound to the glycerol. Triglyceride. So the tri means 3 fatty acids on the glycerol. And this molecule is unsaturated because it has 1 double carbon bond, even though all the other carbon bonds are saturated. Triglycerides are the body's richest energy source. It's stored as adipose in animals. You can just look down at your belly and you'll be seeing triglycerides. It is the most common lipid in our body and the most common dietary lipid when you're eating animal products, meaning an animal fat, like in a steak or in butter. It's the most common lipid. When you're having a diet that is only consisting of simple plant oils like corn oil or something like that, you wouldn't see triglycerides. So triglycerides are mainly found in animal products, but there are some, I think, found in plants. I'll have to look that up. I don't remember. I know cholesterol is not found in plants. I'm not sure about triglycerides. All right, any question about the simple lipids? If not, let's move on to the phospholipid, which I guess is a complex lipid. And it is the main molecule in cell membranes. And what you do is when you get phospholipids together in water, they tend to line up like this, And that also is what our cell membrane looks like, where we have the phosphate portion of the phospholipid facing the inside of the cell, and then the fatty acid portion facing in on each other in a bilayer, because the fatty acids don't like to be around water. And then the phospholipids are facing the inside of the cell because the cytoplasm is mostly water. Any question about any of that? So phospholipids have a phosphate group, and you'll note that this molecule is polar. It's even more than polar. You see, this phosphate group has a charge on it, so this molecule is actually ionic as well as polar. And this group here can dissolve in water, because this part of the molecule is polar, which we call the head. And then the tail, this part of the molecule containing the fatty acids, is nonpolar, and it can dissolve in lipids like oil. So phospholipids are an unusual molecule because they can dissolve in both water, this part, and in oil, this part here. And this makes the molecule biphasic, being capable of dissolving in both water and lipids. There's only a few molecules like that. You do know the other one. Soaps and detergents are like phospholipids, meaning they're biphasic, and they can dissolve in both water, the polar group, and then the nonpolar group dissolving in oil. And that's why soaps and detergents help clean our clothes, because they can remove water-soluble molecules from our clothes and then oil-soluble molecules from our clothes. Any question about any of that? Now, I did mention that phospholipid is the main molecule in our cell membrane. It is not the only molecule in our membrane. Animal cells have a cholesterol molecule in the cell membrane, and that's because animal cells don't have a cell wall, and the cholesterol adds support and what's the word I'm looking for? Stability, strength. It adds strength to the cell membrane. And we'll talk about cholesterol in just a little bit. Any question about phospholipids? Okay, as I stated, when you get phospholipids together in water, the fatty acids come together in a bilayer like this, so that the fatty acid portions will be away from the water. So the water will be on this side and on that side. And then the phospholipids will actually form a liposome, or a micelle, where we'll have the water on the outside of the cell and the fatty acid portion on the inside of the cell membrane, and then the phosphate portion of the phospholipid on the inside next to the cytoplasm. And the liposome is the structure that cells take, meaning this should remind you of a cell. And it is a cell, like in the blood, for example, with the cytoplasm being mostly water, and the head of the phospholipid facing the cytoplasm, and the fatty acid portion being together in a bilayer of the cell membrane. And then the fatty phosphate head facing the outside of the cell, which in blood, for example, will be mostly water. And so the head is facing the water. Any question about any of that? Any question about phospholipids? If not, let's move on to sterols and steroids. A steroid is a lipid with four interconnected rigid carbon rings, meaning 1, 2, 3, 4, and it's called rigid because the molecule doesn't change shape or bend. And this four connected carbon ring is characteristic of a steroid. If there's an OH group attached to the steroid, like right here, we then call it a sterol. And cholesterol is a sterol. Cholesterol is an important molecule in the plasma membrane of animal cells, giving strength to the cell membrane. And cholesterol is only found in animal products, like food of animals. You do not find cholesterol in food or products coming from plants. All right. Any question about steroids or sterols? If not, I've got a question for you. Are all lipids composed of monomers? Hello?
I'm going to say no.
Yeah, the cholesterol and the steroids and the sterols are not. They don't have a monomer. What lipids do have monomers? Okay, remember back what lipids we talked about? Phospholipid was the one. And then simple lipids like the triglycerides are other lipids. Which of these have monomers?
The triglyceride?
Yeah, the triglyceride has monomers. It has a monomer of a glycerol and then 3 fatty acids. So this is a, well, this is a monomer. That's a monomer. That's a monomer. That's a monomer. And together they make the lipid. I don't want to call that a polymer because it's only four parts. And the phospholipid also has monomers. It has the phosphate group and then 2 fatty acid monomers. So lipids do have some of the lipid molecules which are made up of monomers, the phospholipids and the triglycerides. And then there are other lipid molecules that do not have monomers. And all of the steroids and the sterols do not have monomers. Okay? And the lipids are just a little different because some of them are made up of monomers and others are not. But all of the others, the carbohydrates, the proteins, and the nucleic acids, they are all made up of monomers. Okay? Okay, let's move on to the third of the biological molecules, the proteins. Proteins are essential in the structure and the function of cells. Proteins are very diverse. You can find proteins as a structural protein found in tissues like bone, muscle, and hair. There's also proteins which are enzymes, and they speed up chemical reactions. There are proteins that act as transport proteins, moving chemicals across the cell membrane. And there are other proteins that act as regulatory proteins, regulating body functions. Hormones do that. Like in children, there's growth factor hormones that help the children grow, and it's a regulatory protein. There are proteins that are responsible for movement. Whenever you move your muscle, that's because of the proteins. And it's also true for a bacteria that's swimming through water. It's swimming because of proteins. So proteins are responsible for movement. They're also responsible for some of the bacterial toxins. Most of the exotoxins, and we'll talk more about that, are bacterial proteins. And then proteins are also antibodies. These are proteins that an organism makes to defend itself against foreign microbes that can invade the body. So antibodies help defend the body. As you can see, proteins have many different roles and many different functions. And they are the molecule, or I should say the biological molecules, they're the one that varies the most. Probably the reason why they vary more than the other biological molecule is that proteins have more building blocks. Like there's a handful of building blocks that make the carbohydrates, and it's the simple sugars. But proteins are made up of 20 different amino acids, and they're the building blocks for the proteins. So they have more different building blocks. Let's talk about the monomer of protein, and these are the building blocks. We call them amino acids. All amino acids have this generalized shape with the central carbon holding everything together in the amino acid. And bound to that central carbon is a hydrogen. On one side of the carbon is an amino group, that's NH2 right here. And that's what gives the amino acid its name, the amino group. And then the other side is a carboxyl group, which gives it the acid part of the name. I'll explain that in a minute. Anyways, the amino group is on one side of the carbon, the carboxyl group is on the other side. The carboxyl group gives it the acid because this hydrogen can come off, and if hydrogen comes off a molecule, we call that an acid. That's why we have this as an amino acid, that hydrogen can come off of here. And then bound to that central carbon is a fourth group that we call the R group, and the R group can vary for each amino acids. So humans have 20 different amino acids, and that means we have 20 different R groups, one for each different amino acid. For tyrosine, the R group is shown here, and you add that for the amino acid tyrosine. All right, any question about amino acids? To get a protein, you just link many amino acids together, one after the other, in a linear chain to form the protein, using dehydration synthesis once again. Let's talk a little bit about the R groups. There are 20 different amino acids. Each one of them has a different R group. The simplest R group is glycine, which simply has a hydrogen atom in that R group right there. Let me see if I can blow this up a little. You don't need to memorize this table. I'm just showing you the different R groups. Some of the R groups can be nonpolar, like this right here and that right there. Other R groups can have polar regions, like on this R group here, the OH is a polar region for this R group, and then on this one here, the NH and the NH here is polar regions, meaning that depending on the amino acid, some of them will be nonpolar, like that one, and others will have mostly polar regions to the R group. So all proteins have nonpolar regions from some R groups, and then polar regions from other R groups, as well as each amino acid has a polar region from the amino group and from the carboxyl group. So all amino acids are polar, and most of them are also nonpolar from the R group. Most of the R groups are nonpolar, but not all of them. Like this one is very polar. That one is somewhat polar. Any question about that? So proteins have both polar regions and nonpolar regions. And I stated that amino acids are linked together one after another. In dehydration synthesis, you just take one amino acid, pull off an OH group from it, and then bind it to another amino acid, pulling off the H, so the water is made in dehydration synthesis. And then this carbon right here bonds directly to that nitrogen right there. And this bonding between the carbon and the nitrogen is uncommon in biological molecules and is actually uncommon in the universe. So we have a special name for this bond. Because really all it is a covalent bond between carbon and nitrogen. But it's unusual, meaning it's kind of rare in the universe. And so we call it a peptide bond because this bond is found in peptides. And when you have many peptides together, if the molecule is done growing, we call that polypeptide a protein. Any question about any of that? So while the protein is growing, it's not really a protein yet until it gets its last amino acid added on, and then we can call it a protein. Before it has the last amino acid on, it is called a polypeptide. Except when it's very, very small and there's only a few amino acids, then it's called the peptide. Any question about any of that? So what's the difference between a polypeptide and a protein?
The last amino acid.
Yeah, if you add the last amino acid on, then it's a protein. Before that, it's a polypeptide. All right, any questions about proteins and the peptide bonds? If not, let's take a look at the levels of protein structure. Proteins can have 4 levels to the protein structure, and they're called primary level, the secondary level, the tertiary level, and the quaternary level. And hopefully this link will work. Ah. Let's go to, let's just do it here. I don't have it, I'll bet. I have to bring my thumb drive back. I don't have the video on. That's a little longer than I want. Maybe that one will be good.
proteins are polymers of amino acids. So in this video, we're going to take a look at how we can combine amino acids into a protein and how different higher levels of structure for a polypeptide chain are possible. We're going to let the beads represent amino acids. The primary structure of an amino acid refers to the sequence of the different amino acids in the polypeptide chain. So here we have a model of a polypeptide chain with different kinds of amino acids strung together. In reality, they're not strung on a piece of wire, they're connected by peptide bonds. The secondary structure of a protein refers to how the polypeptide chain is twisted on itself. One type of secondary structure is called an alpha helix, and an alpha helix results when the chain coils in a helical structure like this. The secondary structure, the alpha helix, is stabilized by hydrogen bonding between nearby amino acid residues, specifically between the hydrogen on the amide nitrogen and a nearby carbonyl oxygen. This kind of secondary structure is common in proteins such as wool and other stretchy type proteins because the alpha helix has room to give. Another common type of secondary structure is called a beta-pleated sheet. A beta-pleated sheet results when nearby linear strands of a polypeptide chain, which may either be within the same polypeptide chain or between different polypeptide chains, line up in a linear fashion. Again, it's hydrogen bonding that holds this secondary structure together when hydrogens on nitrogens in one chain form a hydrogen bond to the carbonyl oxygen in an adjacent chain. This kind of structure, the beta-pleated sheet, is common in proteins such as silk. And you know from experience that silk doesn't have much give, and this is a physical manifestation of the fact that the polypeptide chains in silk are already as extended as they can go. So secondary structure includes the alpha helix and the beta-pleated sheet. Tertiary structure refers to how the polypeptide chain that has been twisted into a secondary structure folds onto itself to form a compact globular structure. So as I fold the various regions of this polypeptide chain into a more compact structure, that represents a tertiary structure. Tertiary structures are held together by hydrogen bonding between amino acids in different regions of the polypeptide. There's also London forces which result from folding the non-polar alkyl side chains in the amino acid towards the interior of the protein. Now what we've seen here is really a great simplification of reality, because while the primary structure or the sequence of amino acids in the polypeptide chain determines all of the higher levels of structure in the polypeptide chain and ultimately the function of the protein, one of the fundamental unanswered questions in physical biochemistry is how can we predict what secondary and tertiary structures are likely to result based on a given primary structure.
In summary, the primary structure of proteins is the sequence of amino acids in the polypeptide chain. Amino acids are covalently bonded together by amide functional groups, also called peptide bonds. The secondary structure is how the chain is twisted or folded. The alpha helix is a coil of amino acids that are held in place by hydrogen bonding. Wool is a stretchy protein that is mostly alpha helix. Another type of secondary structure is the beta-pleated sheet, which consists of extended chains of amino acids. The beta-pleated sheet is held in place by hydrogen bonding between groups on adjacent chains. Silk is a non-stretchy protein that is mostly beta-pleated sheets. Tertiary structure is how the twisted chain is folded into a compact structure. Tertiary structure is held in place by hydrogen bonding and by London forces among nonpolar groups that are folded into the interior of the protein away from the water environment.
Okay, any questions about that? If you go to my web page, you'll have a, let's turn that up a little. You'll have a different movie, which I think is a little better than that one. I'm not sure why that isn't running here. Ah, here we go. Let's see if that works. No, we're not going to watch another movie, but I'm just trying to get it. Yeah, that is working. All right. Oops. Nope, it's not working. Anyways, let's talk about the different primary, secondary, tertiary, quaternary structure. The primary structure is the simplest and the easiest to remember. It's simply the specific sequence of amino acids in a polypeptide chain, one after the other. So here we got the red amino acid, or orange, followed by the blue, followed by the yellow, and that is the primary sequence, or the primary structure of the amino acid. And when a protein is made, all it has is the primary structure. But shortly after it's made, the nearby amino acids start interacting with each other. And that gives rise to the secondary structure. Now in the video, it said that the main force of the secondary structure is that some of the amino acids can hydrogen bond to each other. And it is true that that is the main force causing the secondary structure in a protein, and that's hydrogen bonding forming between nearby amino acids, but it's not the only force that's giving rise to secondary structure. Secondary structure comes about by the interaction of nearby amino acids interacting with each other. Sometimes there can be attraction, such as this hydrogen bonding right here, but sometimes it can be an ionic attraction, like this amino acid can have a positive charge. Well, that would be a bad example because they're the same. Let's say this amino acid right here can have a positive charge, and that one a negative charge, and then these two amino acids be attracted to each other. That happens more often than you think, because remember I said on the carboxyl group, the hydrogen can come off. Well, frequently it will come off and add to the amino group on the same amino acid or another amino acid. And if the amino group adds here, this amino acid will be positively charged, and this one will be negatively charged, and then they'll have ionic attraction between each other. Any question about any of that? So you can have hydrogen bonding, you can have ionic bonding. You can also have repulsion. For example, these two amino acids may be repulsed from each other. That could happen if this is a positive charge and that's a positive charge. They're going to be repulsed from each other. They can also be repulsed if this is a polar amino acid and this is nonpolar. And then one that they did talk about, but only in tertiary structure, and it can happen in secondary structure, and that is, let's say this amino acid is nonpolar and that amino acid is nonpolar. Nonpolar molecules like to come together. The video call that I think London attraction. I've actually never heard of that one, so I'd have to look it up. I've heard it called van der Waals forces attract nonpolar molecules together. And that's why when you add oil to oil, they come together by the van der Waals forces. Any question about any of that? The point is, secondary structure happens by nearby amino acids being attracted to each other or being repulsed from each other. And that gives rise to a secondary structure. And you don't need to know the different types of secondary structure, but it can be an alpha helix shown here. And I think that's an alpha helix shown here also. It can be the beta pleated sheet shown here. And then the third is a random coil. 3 different types of the secondary structure. You don't need to know those different types. Just know the nearby amino acids are interacting with each other, being either attracted or repulsed from each other. Now, tertiary structure has the same forces of attraction and the same forces of repulsion as secondary structure. The difference is the tertiary structure has both nearby amino acids being attracted to each other, which is actually really secondary structure, as well as amino acids from all over the protein. Like this one's at the end of the protein, and this one is near the beginning of the protein, and they're being attracted to each other. And that's the tertiary structure, where amino acids are interacting with each other, and these amino acids can be anywhere in the protein, not just nearby amino acids. Another difference of the tertiary structure is that disulfide bonds can form between, let me blow this up so you can see it a little better, between 2 sulfhydryl groups that happen in the amino acid cysteine. Cysteine has a sulfhydryl group on it, and this is another cysteine. Well, the hydrogen can come off of the sulfhydryl group, and then the sulfur can bind to that sulfohydryl group, causing a disulfide bridge. And that is a covalent link between this cysteine and this cysteine. That is a really strong bond. An example of how strong the disulfide bridge between cysteine is when you get your hair permed. That creates disulfide bonds between the cysteine of your hair. And then that hair curl will last a month with constant showering. The difference is a hydrogen bond, like when my mother used to curl her hairs in a curling iron, only created hydrogen bonds between the hair. And the first time she would go out in the Oregon mist or rain, because she lived in Oregon, or the Western Washington mist or rain, the hair will uncurl because all it is a hydrogen bond. The point is that this disulfide bridge is a really strong link in the tertiary structure. All right, any question about tertiary structure and how it differs from secondary structure? Most proteins only have a tertiary structure. Like most enzymes, only go up to a tertiary structure. The tertiary structure is the final folding pattern of the enzyme. But some proteins have a 4th structure on top of the tertiary structure, and that we call the quaternary structure. Let me shrink this down. The proteins that have the quaternary structure are proteins that are made up of 4 different polypeptides to make the final protein. So if the protein is made of more than 1 polypeptide, there will be a quaternary structure, like hemoglobin is an example of a protein that has quaternary structure. The quaternary structure has the interaction between amino acids in one of the polypeptide interacting to the other amino acids in other polypeptides. And then when the final quaternary structure happens, when each of the polypeptides come together, that's when we call it the quaternary structure of the protein. So only a few proteins, I don't know how many, have a quaternary structure, and that's a protein that has more than 1 polypeptide. Antibodies, hemoglobin, insulin are all examples of proteins that have quaternary structure, where the amino acid, I can't even see, of this polypeptide is interacting with an amino acid on this polypeptide. The bonds that form between the amino acids are the same we saw in the secondary and the tertiary structures. All right, any question about the levels of protein structure? Not hearing anything? Is there someone there? Yep, we got people there. All right, let's move on. A little bit more about proteins. Proteins have 3 different shapes. They can be globular proteins, and these are compact, roughly spherical proteins. Hemoglobin is a globular protein. Or the protein can be a fibrous protein, that means thread-like. And collagen is a fibrous protein. You can find fibrous protein in some structures like bone and hair. And then globular protein, all enzymes are globular proteins. Most other proteins tend to be globular proteins. As I stated, proteins are more complicated than the other of the biological molecules. They play different roles in cells. Proteins have another complication, and that is they can be conjugated to other molecules. We can conjugate a protein, meaning combinations of amino acids with other organic or inorganic components, and we call these conjugated proteins. They're named by their non-protein component. You probably heard the term glycoprotein. That's a protein bound to a carbohydrate. And we have these in our blood and on our cell surface molecules. Lipoprotein is a protein bound to a lipid. And we have these in our blood. All right, any question about conjugated proteins? As I stated, they are more complex and diverse than the other 3 biological molecules, and they play many roles in an organism or in a cell. Let's move on to protein denaturation. As you kind of learned in the movie on the levels of protein structure, that the folding and the binding of the protein determine its function, its structure. So all proteins will fold into a specific pattern. I don't know if you can see my hands, hopefully you can, which is the structure of the protein. And if it folds into a different pattern, the protein can become denatured, where the protein loses its three-dimensional shape, breaking hydrogen bonds, possibly forming other hydrogen bonds that it shouldn't have. And if the protein changes shape too much, the protein will no longer function. And you can denature proteins by putting them in their incorrect temperature, incorrect pH, or incorrect salt concentration. A classic case of protein denaturation is egg white, that is protein, and when you cook it, you denature the proteins, giving the cooked egg white. That denaturation cannot be reversed. Depending on the denaturation, it could be reversed. An example would be when you take an enzyme and you put it in the refrigerator, the protein will denature, it can't function if it's an enzyme that needs a temperature above refrigeration. But then you can take the enzyme out of the refrigerator, the denaturation can reverse, and the protein can take on its correct folding pattern, and then the protein, the enzyme, can function. All right, any question about any of that? Any question about proteins in general? If not, we're moving on to the next and our final of the biological molecules, nucleic acids. There are two nucleic acids, DNA, which stands for deoxyribonucleic acid, and the second is RNA, which stands for ribonucleic acid. Both of these molecules, meaning both DNA and RNA, consist of monomers called nucleotides. And here we're looking at a nucleotide. A nucleotide consists of a 5-carbon pentose sugar, which is always deoxyribose in DNA and ribose in RNA. And that gives it the name ribonucleic acid, or DNA deoxyribonucleic acid. The nucleotide, besides the 5-carbon pentose sugar shown here, has a phosphate group shown here, and this phosphate group does have a charge on it, so the nucleic acids are a polar molecule as well as a charged molecule. And all nucleotides have a nitrogen-containing base, which can vary. And this is one of the bases, adenine. And all you need to really know is it's a nitrogen-containing base. I suppose you should know that DNA has 4 different bases, so the DNA has 4 different nucleotides. RNA also has 4 different bases, and it has 4 different nucleotides. 3 of them are the same, but 1 differs. We'll talk about that more. The nitrogen-containing base can be a purine or a pyrimidine. Adenine is a purine. Isn't that what I just showed you? The purines of DNA are adenine, or A, and guanine, or G. And you need to know one of the names. You don't really need to know both of them. But if you want to be simple, you just call it A and G for the purines. And for DNA, there's C and T for cytosine and thymine. And those are the 4 nucleotides of DNA. Now, RNA has 3 of the same nucleotides. It does have A, and it does have G, and it does have C, but RNA does not have T. The T is replaced by uracil in RNA. Any question about any of that? All right. For DNA, it's very easy to remember the pyrimidines from the purines because pyrimidine has a Y in the name, and all of the pyrimidines, cytosine and thymine, have a Y in the name. The adenine and guanine do not have a Y. Now for RNA, you can remember this rule, and then just remember that thymine has a Y in it, so it's a pyrimidine, and it's replaced by uracil, which is a pyrimidine. So the rule doesn't quite work with RNA. Uracil doesn't have a Y in it, but uracil is a pyrimidine. All right. The bases bind to each other via hydrogen bond. The A and the T hydrogen bond to each other, and they do so twice. So A and T, this is thymine or T, this is adenine or A. They bind together with 2 hydrogen bonds. C and the G are complementary to each other, and they also bind to each other, but they bind 3 times. 3 hydrogen bonds between them. And let me see if I've got that anywhere. I do not. Let's come down here. Between the C and the G, there's 3 hydrogen bonds. Between the A and the T, there's 2 hydrogen bonds. So that means that A is complementary to T. It will always bind in one strand of DNA to the T in the other strand of DNA, and the G always bonds to C, and vice versa. C binds with G. And that is how DNA, the double strand, holds together, is that the hydrogen bonds between the A and the T and the C and the G hold one strand together with the other strand. So between 2 DNA molecules, they're only held together by hydrogen bonds. In one strand of DNA, it's held together by covalent bonds. And as you know, covalent bonds are much stronger than a hydrogen bond. So does that mean that in DNA, that this bond, or these 2 bonds, break every once in a while in your cells? That's a yes or no question, people.
Yes.
Yes, it does. And that's called the DNA breathing. That your DNA, every once in a while, will break this bond and then come apart. And it's not a problem because the bond can reform and then it'll come together. But in your cells, because your cells are at 37 degrees Celsius, which is 98.6 degrees Fahrenheit, the DNA is constantly breaking those hydrogen bonds and then reforming them. And they call that DNA breathing. I should mention that DNA is a double-stranded molecule, with one strand being held together with covalent bonds, and it binds to the second strand only with hydrogen bonds. So DNA is double-stranded. What's the sugar for DNA? Does anyone remember? Come on, look it up if you don't remember. The sugar is deoxyribose. Okay. The bases are A, T, G, and C. And then the DNA molecule twists like a twisted ladder or a twisted staircase. And what's holding the DNA together, at least in the single strand, is the sugar phosphate backbone. Let me see if I can show that. Oh, this is probably the best. So the sugar phosphate is here, and it connects to the sugar phosphate here, connecting to the, let's see, phosphate, sugar, one after the other. The nitrogen-containing bases are the steps on the stairs, and they bind to the hydrogen bind to the other DNA strand, meaning that the nitrogen-containing bases are not holding the single strand of DNA together. All right, any question about DNA? If not, let's see if we can finish on RNA. RNA, what's the sugar in RNA?
Ribose.
Yeah, the sugar is ribose, and it's a single-stranded molecule. Its molecule is held together by the sugar phosphate backbone, the same as DNA. It's just a single-stranded molecule. And the bases are adenine, guanine, cytosine, and uracil. There are three major kinds of RNA: transfer RNA, called tRNA, messenger RNA, called messenger RNA, and ribosomal RNA, called rRNA. What's the difference between these molecules? Do they have the same nucleotides?
Yeah.
Yes, they have the same nucleotides. The only difference between tRNA and messenger RNA is the sequence of the nucleotides. The arrangement of the four nucleotides, A, U, C, G. That's what only determines whether it's a tRNA, a messenger RNA, or an rRNA. All right. If there's no further questions, I think I'll stop right here, and I'll see you at 6:30 in the lab.
Thank you.
All right.